We have other questions? Glenn.
See, if you want a question, just cite somebody specifically in the audience. This is a good way to do it.
Just a couple interesting comments on recent papers. One by Joe Zabner's group in Iowa, where they moved the human paraoxynase gene into Drosophila and showed increased resistance to pseudomonads, presumably by the quorum sensing factor hydrolysis ability of PON1. So that plugs it again into the innate immunity like the
PON1 is carried on HDL, so that's right. Yeah.
So it plugs it into the innate immunity again like the trypanosome lytic factor.
And Drosophila is a fly model that we use a lot for model system studies. So the fact that you can put a protein into a fly and it kills bugs implies pretty strongly that it's a broad biological function. So these are the kinds of things that we're thinking about.
The other set of observations comes from Jordi Camp's lab in Spain. I don't know if you saw their paper, where they looked at the immunolocalization of PONs 1, 2, and 3 in the mouse, and they found distributions of the of ApoA1 around the mouse system where the A1 didn't co-localize with PON. And so one interpretation would be that HDL sort of serves as a delivery truck to deliver PON1, for example, and perhaps other proteins around the system. Right. It's quite like carrying lipid, but carrying proteins instead.
I can only say that we completely agree. So we've used antibodies to pull out certain other proteins. One that we're interested in is called phospholipid transfer protein. So this protein, again, is generally believed to play a role in transferring phospholipids between lipoproteins, but its proposed crystal structure just looks like a major neutrophil protein called BPI, bacterial permeability inducing factor, which is a major antimicrobial protein in neutrophils. So it looks like a protein that plays a role in host defense mechanisms. When we isolate these particles using antibodies, and this is work that was done in collaboration with Marion Chung and John Albers, we find almost no lipid in the particles and a whole host of proteins that are involved in complement activation and coagulation. The stoichiometry is very interesting. So there's about, again, clusterin comes into the story. So there's about 8 moles of clusterin, 1 mole of PLTP, 1 mole of ApoA1, and then 1 to 2 moles of a wide array of other proteins that are in these complexes. And when we used a database system that predicts interactions known on documented interactions in the literature, we could show that there were documented protein-protein interactions between every single protein in these complexes. So we're very enamored of this idea that protein-protein interactions are really what's responsible for driving HDL. One of the little problems with all this is ApoA1 may not be the whole story. And so there are HDL particles that are known not to have ApoA1 in them, for example, ApoE. And so it gets to be a complicated story. But we think that it's important to deal with the complexity rather than to scramble them into a random mixture of particles and then call that HDL.
A lot of the discussion, both the medical and lay press discussion of heart disease and lipids, is focused on what are called risk factors and things that you or I do that might modify our risk of developing heart disease or stroke. And I guess there are 2 related questions here. One question is, are there common genetic differences between people in this room, for instance, that strongly influence either the nature of HDL particles or their activity? And conversely, how do the things that we think of as risk factors and that there is some evidence for influences our risk of cardiovascular disease alter these multifunctional HDL particles?
So those are great questions. We really know very little about that. We do know that ApoA1 is an incredibly potent risk factor. So there are humans that lack ApoA1. So they have HDL. They have a normal HDL cholesterol if you do the standard blood test. But they have no ApoA1 in their HDL particles. And there's only been 7 or 8 of these families described, but all of the males in the families develop heart disease in their 20s. And we can recapitulate this in mice by knocking out ApoA1. So we can make mice get extensive atherosclerosis with completely normal levels of HDL cholesterol. So there's very strong evidence from animal models that ApoA1 is important. But it's not that simple. I think this issue of the torcept that increased HDL cholesterol but may be bad for you shows that it's not just a matter of measuring HDL ApoA1 cholesterol ratios. We think it's more complicated than that. We're just beginning to try and dissect out what some of these interactions may be, but really virtually nothing is known. There have been a number of genome-wide association studies that have looked for other things that regulate HDL cholesterol levels, and really CETP comes up over and over again. This is a protein that transfers cholesterol esters between particles, but it has a very modest effect, and it doesn't obviously have a major impact on cardiovascular risk. So I think we're just beginning to try and understand what's going on there. But again, a very interesting idea would be, for example, to do a large study looking at SNPs for SNPs that are associated with cardiovascular disease, and to look in parallel in large numbers of individuals at the HDL proteome and to try and make connections there between influences, genetic influences on the HDL protein composition and cardiovascular risk. To do those kinds of studies, we're going to need high throughput assays where we can do a lot of analyses, because you have to have a lot of statistical power to do that. So I think those are great questions, and we just don't know. We have been throwing HDL from mice onto macrophages, and we can control the inflammation in mice experimentally. And what we're finding is that what we call normal HDL or control HDL and inflammatory HDL have very, very different effects on macrophages. And this does not involve cholesterol metabolism. So we haven't figured out yet what it is. It could be the proteins, it could be the lipids, it could be both. But at least in our hands, in mice, inflammatory HDL and HDL have very different effects on inflammation in macrophages, and I think that would be another very interesting area to pursue. Yeah.
Thanks again, Jay.